Hey there, I'm going to walk you through how to complete this meiosis activity. So make sure before you get started that you have both copies of the activity sheet. So you should have meiosis 1 and you should have the meiosis 2 papers, and then you should also have your bag of pipe cleaners and the beads that go with them. So in this activity, you are going to be modeling these stages of meiosis. So you're going to start in prophase 1, and I believe you're going to start with 2 chromosomes, so 1 of each color, okay? And then you need to make sure you go through DNA replication so that you have 2 double chromosomes. So the way you're going to do that is by using your bead to represent a centromere. So here's 1 double chromosome and then a second double chromosome. So I still have 2 chromosomes, but now they have been replicated. Now I'm going to head to metaphase 1. So I'm going to get a whole new set of pipe cleaners. I'm going to line my chromosomes up in the middle because this is metaphase. So now they're lined up in the middle. Anaphase 1, so new set of DNA. So anaphase 1, your homologous pairs of chromosomes are going to move away from each other. So my purple chromosome went off to this side of the cell, and then my blue chromosome is moving in the opposite direction, so anaphase away. So as you can see, I am still representing each of the pairs of DNA in each cell, so I'm going to be using all of my materials here, okay? So here's meiosis 1. Now you would have these stacked, like it says, you've laid out this sheet above the meiosis 2. I have limited space right here, so I'm just going to slide this one over. But at the end of meiosis 1, right, we start in meiosis 2. So I will have 2 cells here that I have to keep track of. So this chromosome, my blue chromosome went off to this cell, and then my purple chromosome went off into this cell. So kind of move over here. So remember, they moved away from each other, right? And now I just have 1 chromosome in each of my daughter cells, but they are still, right, the replicated chromosome, okay? Because in meiosis 1, only the homologous pairs of chromosomes are pulled away from each other, okay? That centromere is still holding the double chromosome together. It's not until the end of meiosis 2, when we get to telophase 2, right, where we will now pull apart our sister chromatids or our double chromosome. So this double chromosome has been pulled apart, and this double chromosome has been pulled apart. So now I have 1 single chromosome or chromatid in each of my 4 daughter cells. Now, at the end of meiosis 2, if you are male, you will create 4 sperm. So this would go here, and these would go here. If you are female, at the end of meiosis 2, you create only 1 egg and 3 polar bodies. So males create 4 gametes for every round of meiosis, whereas females only create 1 gamete for every round of meiosis. These other 3 cells, the polar bodies, are trash. They are not used in sexual reproduction. But as we can see here, I'm going to try to slide this over as best as I can. Right, remember our original cell, we started with 2 chromosomes, and then at the end of meiosis, right, we have 1 chromosome in each of our gametes or our sex cells. So the number of chromosomes has been cut in half, right? So gametes are haploid, and this is important because these haploid cells will go on in sexual reproduction to combine to give us our full diploid number again. So this is the activity, again, for the stages of meiosis. If you have any questions or concerns as you're going through this activity, or if you need any further explanation, please just make sure to let me know.